So let's talk about the metamorphosis in insect. And in particular, let's talk about the metamorphosis in holometabolous insect. So what are holometabolous insect? Holometabolous insect are insects that lay eggs, like Drosophila. From these eggs hatch a larva, and the larva go through several molting stages, producing different instant larvae, like Drosophila will do. Then at one point, the biggest of the larvae, so the last instant larva, undergo a metamorphic molt, so they produce a pupa, and inside the pupa they undergo metamorphosis. Inside the puparium, the larva basically produce all the adult structures, and then we have an imaginal molt where the adults come out eclode from the puparium. So how holometabolous animals, insects, undergo metamorphosis, sorry. Well, in this holometabolous animal, the larva body is almost completely destroyed during metamorphosis by apoptosis, and only some cells called the imaginal cells remain, and these imaginal cells produce the adult structure. Now, we encounter the imaginal cells really when we dissect the larvae to collect imaginal disc cells. The imaginal discs are indeed made of imaginal cells. There are three types, in fact, of imaginal cells that we can recognize in the larva. You can see them here in this cartoon. So we have imaginal disc cells. The imaginal disc cells, we encounter them when we dissect the larva, the Drosophila larva. So we have different imaginal disc cells that produce the cuticular structure of the adult, like the wings, the legs, the antenna, the eyes, the head, the thorax, and genitalia. We have also histoblasts. Histoblasts are imaginal cells that produce the abdomen of the adult. And we have also clusters of imaginal cells that are distributed in all of the larva organs, and during metamorphosis those clusters of cells remain and they produce the organs of the adult. But what's happened during metamorphosis in the imaginal cells that are part of the imaginal disc? So here on this cartoon, sorry, this is actually a picture, an electronic picture. You see one of the imaginal discs. It's very similar to the one that we isolated in our practical during the dissection of the imaginal disc in the larva. You can see that the epithelium, if you want, of the imaginal disc is organized as a spiral. So it is like a tube organized as a spiral. During metamorphosis, the cell proliferate. So most of the cells of the animals, of the insect, do not proliferate much. However, the imaginal disc cell, and in particular, and the imaginal disc in general, proliferate a lot during metamorphosis. And one of the things that's happened is that at metamorphosis, these imaginal disc cells proliferate a lot and they telescope out. So the spiral telescope out. So the middle part of this spiral move outwards. So in this cartoon here, we have an example of what's happened to one particular imaginal disc, which is the imaginal disc for the legs. But very similarly, the other imaginal disc develop in a similar way. So what's happened here is that during metamorphosis, the cells of the imaginal disc proliferate, and the cells in the middle start to move out. So here is a vision from the top. On the second picture, here is a vision, a lateral vision, so a section. While these structures are telescoping out, they form different parts of the organs, in this case the leg. The central part of the spiral telescoping out will become the most distal part of the organ, in this case the claw and tarsus, while, while for example the most outer part of the spiral will become the most proximal part of the organ, in this case the coxa and the trochanter. But what genes are expressed in different parts of the imaginal disc that are associated with developing the different part of the organ? And again, here we are using the example of the imaginal disc for the leg. First of all, the specific genes that are expressed in a different part of the imaginal disc are controlled by the OX genes. OX genes control the expression of the imaginal disc and cells. For example, ultrabithorax and actinopedia are active in the imaginal disc of the legs. Now, in the center of the imaginal disc, by the third instant larva, in the center of the imaginal disc, we have expression of DPP and wingless. Now, DPP and wingless are a potent transcription factor that are expressed in the center of the imaginal disc, and they diffuse outwards from the place of expression. High level of DPP and wingless induce the expression of distantless. And here, in this picture here, you see in red the region of expression of distantless. Distantless is expressed in the center of the imaginal disc. And then you can see here the structure of a leg, of a formed leg, and you see the distantless is expressed in the most outward, if you want, the most distal part of the leg. So you see that the cells that express distantless, which are on the center of the spiral or the imaginal disc, now are located more distantly in the structure in the leg. Moderate level of DPP and wingless are necessary for expression of Dutch hand, and you can see the expression of Dutch hand here in green in the imaginal disc, and here in the leg that is formed from the imaginal disc. And the region that express Dutch hand correspond to the femur and proximal tibia. Low level of DPP and wingless are required for the expression of homothorax. And homothorax, you can see expressed here on the edges of the imaginal disc, correspond to expression on the coxa of the leg, which is the most proximal part of the leg itself. And finally, an overlap of expression of Dutch hand and distantless cause expression is necessary to express the trochanter. Trochanter is and the distal fibia are the middle part, if you want, of the leg. So a gradient of expression of DPP and wingless from the center to the periphery of the imaginal disc will cause expression of different other transcription factor, which specify the expression of specify the formation, if you want, of different part of the leg of the insect. This kind of organization is also repeated in other imaginal discs. Now, what hormones control metamorphosis and molting in general? Well, the hormone that controls molting in insect is 20-hydroxyectisone. For simplicity, I just call it ectisone. And we have several pulses of ectisone are necessary to initiate pupil formation and then to drive metamorphosis itself, so eversion of the different tissues. How excuse me, how metamorphosis is controlled hormonally in insect? There are two main hormones that are important for metamorphosis in insect. One is ectisone and the other one is juvenile hormone. Now, ectisone initiates each molt and organize gene expression, so regulate gene expression. Juvenile hormone, on the other hand, prevent ecdysone to induce a substantial change in expression from larva to pupa. So if we have expression of ecdysone and we have expression of juvenile hormone, the only molt that can happen is larva to larva, because juvenile hormone, if you want, maintain young the individual. So it doesn't allow ecdysone to shut down larval genes and turn on pupa genes. So the molting, of course, it is initiated in the brain. Why is that? Because the brain acquires all the information, being environmental or internal information, that are required to instruct the initiation of a molting process and even a metamorphosis process. So if the brain instructs the protothoracic gland, the brain produce protothoracic hormone, which instruct the protothoracic gland to express ecdysone. Ecdysone is the molting signal. Now, the PTTH induces the expression of ecdysone. Ecdysone will instruct the formation or the initiation of molting. Now, the corporalata, which is another gland in the insect, produce juvenile hormone. So if the corporalata produce juvenile hormone, then ecdysone can only produce larva to larva molting. However, if juvenile hormone is not produced, then 20 ecdysone or ecdysone initially induce a prepupal morphogenesis, and in doing that it stop the expression of larva specific genes, and then secondly a second pulse of ecdysone will completely shut down the larva genes, but also activate the pupa and prepupa genes. So the first pulse of ecdysone without juvenile hormone will shut down the larval genes, will start to initiate the pupa and prepupa genes. A second pulse of ecdysone will consolidate the expression of the pupa genes, and therefore the animal will go from a larva to a pupa. So it starts metamorphosis. Now, 20 ecdysone, or ecdysone for simplicity, need to have access to the DNA, of course, to switch on or off different genes. Now, to have access to the DNA, 20 ecdysone need to bind to the ecdysone receptor, or ECR. ECR actually form a dimer with another protein, which is called ultraspiracle, USP. And this ultraspiracle with the ECR already sit on promoter and enhancer of target genes. So ECR, the ultraspiracle, repress the transcription of the target genes. And only when ecdysone arrive and bind the ecdysone receptor, these target genes can be expressed. And actually, we can see the expression of these target genes because of the formation of this path of DNA. So the DNA actually open up and it becomes transcriptionally active, and we can see them under microscope using the salivary gland chromosomes of the flies. Now, if you recall, a similar situation was found also during metamorphosis of amphibian. In amphibian, we have the thyroid receptor that was binding the RXR, and they were sitting together on target genes repressing their expression. But when the thyroid hormone comes, the T3 comes, then those target genes become active. Similar here, we have the ECR, the ecdysone receptor, with ultraspiracle sitting on target genes and repressing their activity. But this target gene become active once ecdysone arrive and bind ecdysone receptor. Okay. So, I hope that after this lesson, after you revise, you will be able to explain similarity and differences between the mechanism, the metamorphic mechanism, both in amphibian and insect. You should be able also to discuss the main morphogenic event that happen in amphibian and in insect, and also the hormonal controls in both insect and amphibian.